Very good morning to all the students. So we have completed Living World, Plant Kingdom, Biological Classification, Plant Kingdom 3 chapters, followed by Morphology of Flowering Plants 4, and then Cell the Unit of Life and Cell Cycle and Cell Division. So totally 6 chapters we have completed. So now next, plant physiology. So the next unit, which is plant physiology. So in this plant physiology, we are going to study about the photosynthesis, respiration, followed by plant growth and development. In plant growth and development, as per the related syllabus, we have to discuss regarding the plant growth regulators. Okay? Right. So these are the different, so these 3 particular chapters we have to study in plant growth and development. If at all, if there is complete syllabus for NTA, as per the NTA, then we will discuss the remaining deleted portion also. So first let us complete the present syllabus as per the PU board. Okay? Right. So the deleted syllabus, what we have to discuss is nothing but the anatomy. Next, followed by the mineral nutrition, and then transport in plants. Those are the topics for the deleted syllabus. Okay, that we'll see later. Now photosynthesis. So what is this important chapter? Photosynthesis, where you come across with different pathways. So cyclic and non-cyclic photophosphorylation, C3, C4, photorespiration. Okay, all these things we have to study. So let us see in detail about the photosynthesis. The word photo means light. Photo indicates light. Synthesis means preparation. So plants prepare their food by using the sunlight and water. We call it as photosynthesis. So which is absent in the case of heterotrophs. So these are called as autotrophs. So note down, photo indicates light. Synthesis, preparation. Preparation of what? Food. So plant prepare their, so what is the definition of photosynthesis? If you see, the synthesis of carbohydrates by plants in the presence of sunlight and water. In the presence of sunlight and water, the preparation of the synthesis of the carbohydrates. So that is what generally we call it as a photosynthesis. Definition of photosynthesis. Next. So these plants, shortly, sunlight and water. By using sunlight and water, plants prepare their food. So how much? So carbon dioxide it utilizes the carbon dioxide, water, and then prepare the carbohydrate. So what is the number of molecules to prepare one glucose molecule? 6 CO2, 12 H2O, water molecules. In the presence of sunlight and chlorophyll. Pigments. So pigments, chlorophyll pigment, which is important chief pigment for the photosynthesis. Glucose, C6H12O6, oxygen and water. Okay, so this is for the photosynthesis. Next, this here water gets oxidized and carbon dioxide gets reduced. Very important. The water gets oxidized. The water gets oxidized. That is, it's a source of proton and oxygen, which is a source of protons and oxygen. Whereas carbon dioxide gets reduced. The carbon dioxide gets reduced. So here, which is the source of carbohydrate? This is the source of carbohydrate. Okay. Then, so here the light energy gets converted to chemical energy. Okay. So what is the light which is taken from the sun? So that light is converted to chemical energy, which is nothing but the carbohydrate. So light energy converts to chemical energy. So light energy is converted to chemical energy. So here, that is the photons, light energy in the form of photons, which is converted to chemical energy, which is ATP. So that is physico-chemical process. Photosynthesis is therefore photosynthesis is a physico-chemical process. As it is converting the light energy, photons, to chemical energy, it is a physico-chemical process. So if you see the photosynthesis rate, 90% of the photosynthesis occurs in the oceans, because 90% of photosynthesis occurs in the oceans, as we have most of the autotrophs like algae, which is present, and lower plants are present mostly in the oceans. So photosynthesis rate is more in the ocean. And another important thing is 1% of light is used for photosynthesis. 1% of total sunlight is used for photosynthesis. So only 1% of the light is used out of the complete sunlight. Only 1% is completely used. Okay, right. So different experiments, early experiments were carried out by different scientists to prove the photosynthesis. So that is variegated leaf experiment, the experiment to prove photosynthesis. Photosynthesis by plants. In the early days, if you see, variegated leaf experiment, variegated leaf experiment and followed by Moll's half-leaf experiment. Variegated leaf experiment and Moll's half-leaf experiment. So these are the two experiments for to prove the photosynthesis by plants. So what are these experiments? Let us discuss these experiments. So keep the heading variegated leaf experiment. Variegated leaf experiment. So what is this variegated leaf? Variegated leaf meaning is it is partially the leaves which are having spots, green and non-color green, non-green. Green and non-green part of the leaf is called variegated leaf. Green and non-green parts, pale color, parts of green and non-green, green and non-green leaf is called is variegated. Is called variegated leaf generally. Okay. So therefore, it is a variegated leaf. So this kind of leaf, if you take and check the rate of photosynthesis where in the green or non-green part, then you will clearly understand this kind of experiment. So here the plant, which is the potted plant, you have to take a potted plant, the potted plant, and the leaves should have both green and non-green, and the leaf should have green and non-green, or if at all they are completely green color leaves you have, or you can partially covered with black paper. Cover the leaf with black paper. So what is the meaning of this? If at all, if the leaf is not non-green, if it is completely green color leaf is present on a plant, then you cover partially half of the leaf with the black paper. Then you can clearly understand that the light cannot even penetrate through it. Okay, right. After that, you have to check whether it is having the green or non-green part. Now the photosynthesis rate where it is present. So such kind of a plant, if you take okay, if you take such kind of a plant and if you see, then this, if you subject for sunlight for few hours, subject the plant, potted plant, subject the potted plant for the sunlight in few hours, then the leaf is collected, then collect the leaf, then collect the leaf, boiled in alcohol. You just collect the leaf and boil in alcohol. You boil this leaf in the alcohol such that what happens when you boil this? It disturbs the cell membrane and softens the cuticle. If any cuticle is present, that it will soften the alcohol, and then the cell wall allows the iodine what you are washing with the leaf, and penetrates to react with the starch, because during the sunlight starch is produced in the leaf. Remember all of you, during photosynthesis the green color part of the leaf will synthesize the starch. So when you add, because iodine test, starch test is nothing but the iodine tester, you boil with alcohol and add the iodine. So when you add the iodine, if you boil it in alcohol and add it, what happens? The alcohol will softens the cell membrane, cell wall. Cuticle is present, cuticle and cell wall. Then it easily allows the iodine to penetrate into the leaf, so that inside the leaf, if you see the starch which is present in the mesophyll cell, that it go and reacts to give the blue color. So even though photosynthesis takes place in the green part also, if you don't boil with alcohol, iodine cannot able to penetrate inside the leaf. So that is the reason you boil it with alcohol, add with iodine. Okay. Then if you test with the iodine, add iodine. So here, then if you see what kind of changes you see, then the green part and non-green part of the leaf, the green part and non-green part of the leaf, this will turn to blue color. This will be colorless. So no, it does not turn into blue. So this is what we see when you check with the iodine. So therefore, what you can understand, whatever the part which is non-green means the part of the leaf which you are covered with black paper. If it is green in color, you are covering with black paper, part of the leaf. Or else if it is variegated leaf and patches of non-green is present, that non-green patches will not turn into blue color. The green color parts of the leaf will turn into blue color. So therefore, by this experiment, what we can infer means the light is required for photosynthesis. Therefore, what is the conclusion for this experiment? Light is required. You can say, therefore, light is required for photosynthesis. Therefore, light is required for photosynthesis, or photosynthesis occurs in light. Photosynthesis occurs in the light. So this is what we call it as a variegated leaf experiment. Is it clear, all of you? Right. The next experiment is moles half-leaf experiment. Moles half-leaf experiment. So what is this experiment says? This is carbon dioxide is necessary for photosynthesis. The second experiment, moles half-leaf experiment. So this experiment says that carbon dioxide is necessary for photosynthesis. These conclusions you have to remember. Why we do this experiment, that you should understand. Okay, that only they'll mostly ask in the exam. Why we do variegated leaf experiment? What is the reason for variegated leaf experiment? What proves the variegated leaf experiment? All these things they will ask. So remember. So what is the main thing, aim of this? The moles half-leaf experiment is to prove that carbon dioxide is necessary for photosynthesis. Carbon dioxide is necessary for photosynthesis. Next, what is this? If you take the against the leaf, whatever the plant leaf, you take a leaf which is exposed to the sunlight, or you take a leaf in a test tube, the leaf half of the leaf is completely kept inside the test tube, and half of the leaf is left outside. Okay, and inside you keep a cotton soaked with potassium hydroxide. The cotton soaked with potassium hydroxide. So what does the potassium hydroxide will do? It absorbs any carbon dioxide which is present inside the test tube. So you cover the openings of the test tube tightly such that no air should pass inside. If any air is present inside, the present air inside the tube will be absorbed by the carbon dioxide. Will be absorbed by the potassium hydroxide. What is the potassium hydroxide will do? The potassium hydroxide absorbs carbon dioxide which is present in the carbon dioxide. So then this part of the leaf you are exposed to air. This is exposed to air. The part of the leaf which is exposed to air, and the remaining part of the leaf is enclosed in tube. Which is enclosed in the test tube. Then you keep this for few hours in light. Expose this for few hours in light. When you expose this for the few hours in light, then what happens? and then you add iodine. Again same thing, iodine tester. When you add iodine, again the enclosed leaf is non-blue color, whereas the exposed leaf will be turned into blue color. Why? Because here what happens when you add iodine, the exposed leaf turn into blue color, whereas enclosed in the tube, part of leaf enclosed in tube, the part of the leaf enclosed in a tube, so that is non-blue. In the part of the leaf which is exposed in the tube, blue color is absent. So here, if you take in the blue color photosynthesis, that is starch is present. Here starch is absent. Starch is present, the starch is absent. So blue color. So here when you see this, I am writing here again, blue color is absent. That means no starch. Here blue color is absent and starch is also absent. So that is why this experiment proves that the carbon dioxide definitely, when it is present in the air, exposed to air means here what is there? Carbon dioxide is present. Inside carbon dioxide is absent. So even though you pass the sunlight to both the parts, see why you are keeping in the test tube? You are not closing, covering with the black paper. Here you are just exposing to the sunlight. The leaf is exposed to the sunlight both inside the test tube and outside the test tube. Is a common. But even though sunlight is present also here, when carbon dioxide is absent, then the starch is not prepared. Okay? So therefore, we call it as the moles half leaf experiment. So moles half leaf experiment proves that carbon dioxide is necessary. Next, early experiments proved by the other scientists. What are the early experiments? Very, very important experiments. The early experiments which are keep the heading early experiments by different scientists regarding photosynthesis. So this is Aristotle. He first defined something regarding this. So the Joseph Priestley, the scientist, different scientist who has done these experiments for photosynthesis. Those are Joseph Priestley, So this is the period of 1733 to 1804. 1733 to 1804. Next, Jan Ingenhorst. Jan Ingenhorst. So this is in the period of 1730 to 1799. To 1799. Next, the Julius von Sachs. Julius von Sachs in 1854. In 1854. Next, Angelman, T.W. Angelman. So he 1843 to 1909. His experiment regarding photosynthesis. Next, Con von Neil. Cornus von Neil. So this is 1897 to 1985. So this is with the early experiments. Rubin and Kamen. Rubin and Kamen in 1941. Rubin and Kamen in 1941. So all these are the different scientists who worked for photosynthesis, who proposed their own ideas and experiments for photosynthesis. So Joseph Priestley, Jan Ingenhorst, Julius von Sachs, Angelman, Cornus von Neil, Rubin and Kamen. So these are the different scientists. Okay, right. Now, so let us see these experiments in detail, what they proposed. So before all these experiments, the Aristotle, he stated initially that the plants absorb the organic, both organic and inorganic compounds are taken from soil by plants. That was proposed by Aristotle. Aristotle initially in the 320 BC, early back, he stated that both organic and inorganic compounds inorganic compounds are taken from soil by plants. Okay, are taken from soil by the plants. So this was a statement given by the Aristotle, but generally we don't see that. They only take the inorganic compounds from the soil and they prepare the organic compounds. Plants take inorganic compounds from the soil, mineral nutrients, and then they prepare the organic compounds like glucose, sucrose, glucose. Okay. So the Joseph Priestley, what he stated. So he has done the candle jar experiment. He has done the candle jar experiment for the essential role, to prove essential role of air. He proved the essential role of air. proved the essential role of air. So, the experiment he carried out is nothing but oxygen evolved by plants, essential role of air, and oxygen is evolved from plants. Oxygen is evolved from the plants, he proved. So this is, the experiment is, he proved with the candle jar experiment. He proved with the candle jar experiment. The candle jar experiment was carried out by the Joseph Priestley in 1772. In the year 1772, the candle jar experiment was proved, and he discovered oxygen in 1774, and discovered oxygen in 1774. The oxygen was first discovered in the year 1774. Okay. So, the candle jar experiment in 1772, what he has performed, that he added. So, what is the candle jar experiment? Let us see the candle jar experiment. In the candle jar experiment, he has, inside the jar, he has kept the candle, burning candle. Okay, and he has kept the mice, the burning candle and the mice he has kept. And when the jar is closed, until the mice survives, when you close it, then the mice gets suffocated. So carbon dioxide, which is released by the mice here, so that until the oxygen which is present, the oxygen is taken by both the mice and followed by it is utilized by the candle. So after few hours, after few hours, the mice died and the candle off. The candle get put off because there is carbon dioxide which increases. Here carbon dioxide will increase because that will be released by the mice. So increase the carbon dioxide leads to the death of the mice, and the candle also will off. But later, when you keep the mint plant inside, burning candle, mice and mint plant, then what happens? Continuously the carbon dioxide which is released by the mice will be utilized by the mint plant, Therefore, this will start release the oxygen, which is utilized by the mice and in turn by the candle. So until the oxygen is continuously present, the mice is alive. The mice is alive and candle is on. The candle is on and the mice is alive. Why? Because the plant itself will release the oxygen by taking the carbon dioxide which is present in the jar. So therefore it proves that oxygen is evolved from the, therefore what can you prove by this? The oxygen is evolved by the plants. So this experiment is that. So essential role, to prove that essential role of the thing, and oxygen evolved by the plants. So here oxygen is evolved by plants. So this is the first experiment which was proved by Joseph Priestley. Is it clear? Right. So the next experiments, in the next class we will discuss the remaining experiments. Those are the other scientists. Okay. Right. Okay. So see you in the next class. Thank you.